Well, hi everyone, and yeah, thanks for staying. We're into the home run now. Yeah, and an honour. I'm really just a spokesman for this fantastic team of investigators you'll see on the slide there. And I think this is going to be the first talk of three on myotonic dystrophy, so it's such an exciting time to be in this field at the moment. So I'm going to be talking about the safety and efficacy of DYNE-101 in adults with DM1. This is the ACHIEVE study. I'm a PI in Manchester in the UK on this study. So here are my disclosures, and obviously important to mention that DYNE-101 is an investigational product which is not yet approved. So myotonic dystrophy, we know it's a spliceopathy. We know that this expansion of the CTG repeat in the DMPK gene leads to these hairpin loops in the RNA that's transcribed from that gene, and that sequesters MBNL. MBNL would normally regulate splicing, so without it you get these missplicing events, impairing normal protein synthesis across multiple genes, across multiple tissues. And the consequences of this spliceopathy are multi-system. It's not just a myopathy, as we all know. Myotonia and muscle weakness, obviously key manifestations, but we've got to think about the central nervous system symptoms and signs, which are often the most troublesome for my patients, including cognitive impairment, fatigue, excessive daytime sleepiness, etc. The cardiac conduction defects, the pulmonary abnormalities, and cardiorespiratory muscle weakness are obviously key considerations as well. So a treatment for myotonic dystrophy is really going to have to address all of these issues and show broad distribution to those tissues. And indeed, that's the goal of treatment with DYNE-101. So what's DYNE-101? There's a little cartoon of it there on the left. You can see it's an FAB molecule with a linker, and then an ASO which gets into the nucleus, and it binds to these hairpin loops, displaces the MBNL that's trapped there, and allows the MBNL to go on and do its thing and correct splicing, which then leads to early clinical effects, improvement in myotonia, which hopefully I'll convince you of with the data I'll show in the next few slides, as well as broad functional improvements in strength and other measures of muscle function. So here's the outline of the ACHIEVE trial. It was a multiple ascending dose study with the various dosing regimens shown there in this cartoon here. I'll be showing data from this cohort, the 6.8 milligram per kilogram cohort dosed every 8 weeks. Participants that were recruited to these lower dosing cohorts are given the opportunity to step up to that higher dose in the open label extension and long-term extension of the study. It's important to note that these are adults with DM1 and that the safety, so the primary endpoint is the safety and tolerability of the medication, although there are some key secondary endpoints including splicing correction, multiple measures of muscle strength and function, and also, importantly, the patient-reported outcome measures including the MD high. Now, this was designed to be a potentially registrational study, and Dyne have gone on to select this 6.8 milligram per kilogram dose given every 8 weeks as the go-forward dose for their registrational expansion cohort, which is just starting to recruit and due to complete by mid-2025. So here are the baseline characteristics for the treated cohort here, and then the pooled placebo cohort on the left. You can see they're pretty evenly matched. Perhaps the only little differences to point out is that the MD high is slightly higher in the treated group, and the 10-meter walk-run and 5 times sit-to-stand slightly longer, suggesting slightly more. Shall I carry on? Yeah. So I think you've all had long enough to look at those baseline characteristics. Now I'll move on. So here's the primary outcome. So remember, safety and tolerability is a phase 1/2 study. In essence, the safety profile is favourable. There were 6 SAEs, but they were deemed unrelated to the study drug, and they're listed out here on the right. The AEs are mentioned there. And it's important to note that none of those were dose dependent. Elevations, abnormalities in liver function tests were seen, although it is difficult to disentangle that. As you'll know, it's quite common to see abnormal liver function tests in patients with DM1 pretty frequently. And there were no abnormalities in liver synthetic function, including the bilirubin and coagulation profiles. And again, no participants showed persistent related anemia or thrombocytopenia. And this data is reflecting 855 doses and 72 patient years of follow-up. I'll just talk briefly about the splicing index. This isn't data from ACHIEVE, this is from the NDM1 natural history cohort. So the splice index, or the composite alternative splicing index, or CASI 22, it really is a measure of the severity of this missplicing. So we're getting to the nub of the cause of myotonic dystrophy. And I think the key thing about the CASI is it changes relatively rapidly. So you see between baseline and 3 months here, there's a significant difference in that CASI. And the other thing is that the CASI correlates with measures of function, and importantly, it correlates more strongly with measures of function at a later date. So these are 3 months, this is myotonia, 10-meter walk run, and some muscle strength. Those correlations are stronger 3 months later as compared to the baseline CASI as time-matched assessments. So it suggests that there's some prognostic power to the CASI as well. So then looking at the DYNE-101 treated cohort, you'll see here the DMPK knockdown on the left, with a robust decrease there of 33% compared to the placebo group there. If you then look at the CASI correction, again a robust improvement there of 25% reduction in the CASI, and then a worsening in the placebo group as you'd expect given the natural history data that I've just showed, and that's at 3 months. If we moved on now to some measures of muscle strength function, and here we have myotonia. So this is the VHOT, the video hand opening time. This is at 6 months now, so a little bit later down the line, out here on the right-hand side of this graph, and you see quite a big decrease in myotonia there in the treated group, reduced by 38% from baseline, compared to a slight worsening in the placebo-treated group. If we now move over to other measures of muscle strength and function, so the total QMT score here on the left, this is a composite measure of 6 different muscle groups. That's improving in the treated cohort, around 10% after 6 months. 10-meter walk run, 5 times sit to stand. So these are measuring things like balance and stamina as well as strength. And you'll see here, as you go down here, that indicates improvement. And we've got a 7.2% improvement in the 10-meter walk run test, and a 12.4% improvement in the 5 times sit to stand. So again, favouring treatment with DYNE-101 at that 6.8 milligram per kilogram dose over placebo, and showing the MD high. So this is a patient-reported outcome measure looking at 17 different domains of health and disease severity in myotonic dystrophy. And I think one of the important things about the MD high is that there's several subscales relating to central nervous system function. And we've seen here the overall total MD high score dropping 44% at 6 months, so a really marked decrease. I think quite a clinically meaningful decrease in the MD high there at 6 months in the treated group. And then if you look at those subscales, and obviously this is small numbers, and we're looking really at trends, but it is really quite exciting to see those improvements across the board on those different CNS-related subscales: cognitive impairment, sleep, fatigue, pain, emotional issues, communication, etc., in the treated group compared to placebo. So I think when you put all this together here, and this is a summary slide of the results, you see that 25% improvement in the CASI at 3 months, then translating to that improvement in myotonia, muscle strength and function, and the patient-reported outcome measures at 6 months. You'll see the blue lines here all towards the right of those grey placebo lines and favouring DYNE-101. So in summary, DYNE-101 targets that nuclear mutant DMPK RNA, releases the MBNL. There's a favourable safety profile demonstrated. It's sort of treating the cause of the disease, correcting splicing, and that's demonstrating benefits, in this small number of patients at 6 months. And this multiple ascending dose portion of the study is now completed, and as I mentioned, the registrational cohort at that 6.8 milligram per kilogram dose is now open and recruiting. And as I say, I'm really just a spokesman for several fantastic investigators across Europe and over in New Zealand. And of course, thank you to the patients and families living with DM1 who surprise me every day with their resilience. So thank you very much for listening. I'll end there.
Today, and a big thank you to those of you in the audience who are hanging out till the end of the meeting, because I know it's getting on and there's been a lot of good science going on, as always, at this meeting. I'm privileged to be presenting on behalf of my colleagues and our investigators to talk to you about PGN-EDO DM1 and our single ascending dose study in people with myotonic dystrophy. Those are my disclosures. We'll go through those quickly. So just a background for those of you who don't know about our EDOs. These are enhanced delivery oligonucleotides, and it's a platform that PepGen is developing to develop a pipeline of disease-modifying therapeutics for neuromuscular and other neurological diseases. Our EDO platform is a PPMO, a peptide-linked oligonucleotide, and those of you who were here earlier for my colleague Paul's talk, he talked a bit about what that was, but the bottom line is Our EDO-peptides allow delivery of oligonucleotide therapeutics, not just into the cell, but into the nucleus. You can see here on the left, untreated cells and cells treated with naked oligonucleotides, and you can see there's no red there. No oligonucleotide is really getting into the nucleus. Here you can see at 2 micromolar and even more at 10 micromolar, lots of red dots showing we are indeed getting oligonucleotide into the nucleus where it's needed to work. I don't need to talk to this audience about what DM1 is, particularly as Diane has just presented on it, but DM1 is caused by a CUG repeat sequence that forms hairpin loops that trap muscleblind and other splicing factors. Unlike some of the other molecules in development, what our EDO-DM1 does is it binds to those repeat sequence, effectively interfering with the hairpin loops and liberating muscleblind without knocking down DMPK. So don't take my word for it, that's what we designed it to do. This is some early cell data that we've had for some time, where you can see the number of foci on the top left are decreased by about 54% when treated with EDO-DM1. We then stain for muscleblind, and you can see liberation of muscleblind from those foci, and that correlates with dose-dependent correction of splicing and myoblasts. I'm going through the preclinical data very quickly because I want to get to the trial results. But this is in the HSA-LR mouse model, where you could see single doses give you a good amount of drug in the tissue. Repeat doses give you even more. These are monthly doses. As you would expect, with more drug accumulating with repeat doses, we see the same with splicing. A good amount of splicing after one dose, even more splicing after repeat doses. And that translates to the correction of myotonia that you can measure in this mouse model. So we were pretty confident going into the clinic that our hypothesized mechanism worked, at least in animal models. So from there, we designed the Freedom DM1 single ascending dose study, which is really what I'm going to be talking to you about today. We've dosed the first 2 cohorts of the study at 5 and 10 mg per kg, and we're currently dosing at 15 mg per kg. And I'm going to share some interim data from those first 2 cohorts. As you can see here, each of the cohorts we're looking at day 28 data, and we actually have a time point out to week 16 because in the mice we saw really sustained correction of splicing. I'm not going to be talking about that week 16 data here, but just know that we will have that data in the future. This is the baseline characteristics of our first 2 cohorts. On the whole, they were pretty well balanced. These are small cohorts. It is a single ascending dose study, but I want to draw attention to a couple of things. Firstly, our splicing index was slightly lower in the 10 mg per kg cohort. That means there were slightly less affected people in that cohort. And probably more importantly, if you look at our VHOT, that was not evenly split between the cohorts. Our placebo had extremely high VHOT. So did our 5 mg per kg and the 10 mg per kg a little bit less. And I'll explain why that's important in a moment. A single ascending dose study is, of course, a safety study first and foremost. Our safety is looking pretty good. We've got a very favorable emerging safety profile so far. We did have one serious adverse event in the 10 mg per kg cohort. One of our participants took an off-label drug the same morning. She got her dose of EDO-DM1. She had some abdominal pain that went away after a few hours. And that, of course, because it happened straight after infusion, is considered drug-related, although, of course, we don't know because of the other drug that she also took. We've had 2 SAEs that were considered unrelated to study drug, a case of appendicitis many weeks after dosing, and a pseudoaneurysm, which was considered related to the biopsy procedure. But importantly, knowing the history of PPMOs, we've seen no adverse events related to electrolytes or renal biomarkers, or really anything else much. My CEO asked me to put this slide in because we always get asked about renal biomarkers. As you can see here, our serum creatinine stayed absolutely within the normal limits in both cohorts. So we study the renal effects very closely, but it's been looking really good so far. Effectiveness. Well, we got the drug into the tissue. You can see here we had dose-dependent increases, in fact, slightly greater than dose-proportional increases in the amount of drug that we got in between 5 and 10 mg per kg. We were quite excited by that. But what we were really excited about is on the next slide, which is that more than dose-proportional increase in drug concentration also translated to more than dose-proportional increases in splicing improvement. So you can see here, at 5 mg per kg, we saw about a 12% improvement in splicing index, and a 29% increase in splicing index. Bear in mind, this is after a single dose, and we anticipate, based on our mouse data, it's going to improve with repeat doses. So we were really quite excited about this. 3 out of 4 people showed an improvement. They showed more than 17% improvement. The fourth did not. That's probably the nature of a single dose, but we're really looking forward to seeing what this looks like after repeat doses. We were a little surprised, but also very encouraged by this data. Functional outcomes, well, after a single dose, not surprisingly, we didn't see a lot. You can see on the left in the 10-meter walk-run test, the 10 mg per kg dose did see a 0.35 second improvement in the 10-meter walk-run time. We don't think that's particularly meaningful at this point, but we are anxious to see how that looks after repeat doses. VHOT, everyone looks great. Our placebo looks particularly good. Our placebo was apparently really good at fixing myotonia. As I mentioned early on, there was some imbalance in the baseline myotonia, and we think that's what happened here. We can't claim any improvement on myotonia after a single dose, given what our placebo did. But again, single dose, we're really looking forward to seeing what repeat doses does. So to conclude on the data we have so far for EDO-DM1, we have a favorable emerging safety profile in people with myotonic dystrophy. We've seen dose-dependent increases in drug concentration across the first 2 cohorts, and we've seen dose-dependent increases in splicing correction following a single dose that are really quite exciting levels of splicing correction, which we anticipate, based on what we've seen in animals, will only improve with repeat dosing. So we're really encouraged by where we are right now. So encouraged, we started our multiple ascending dose study in parallel with finishing our single ascending dose study, and we're currently dosing our first cohort in the repeat dose study at the moment. And I'm hoping by next year we'll have data to report on repeat doses to see if our mice are predictive, and we do in fact see even better splicing improvements and functional improvements after repeat dosing. So with that, I just want to say thank you, particularly to our PIs who are running the study, the participants in the study, and also all my many colleagues who work extremely hard. As you can see from the photo, we're a very small company. We don't have a lot of people. Everyone's working incredibly hard. There's more information, both non-clinical data and clinical data, on those posters, which you can look at electronically. And as always, a big thank you to the whole community who make what we do possible. I'm happy to answer any questions.
As I mentioned, I'm Emily Fantelli. I work with the AMO Pharma team on the clinical development team. And today I'll be presenting the long-term safety, tolerability, and efficacy of AMO-02 in children, adolescents, and adults that have the congenital and childhood version of DM1. So I'd like to thank MDA for the opportunity to present these data today. I don't need to spend much time, thanks to Dr. Lilliker and the PepGen presentation talking about DM1. However, the majority of our research has been focused on the congenital and childhood onset forms of DM1, and it's just important to note that there are phenotypic differences as we go through whether or not someone has the congenital form and has it at birth, or the symptom onset comes on during childhood or has more of an adult onset presentation. However, the clinical features can be a bit across the board with the myotonia, the muscle weakness, fatigue and sleepiness, cognitive problems, and GI issues. And certainly in the kids, we see a bit more of the cognitive problems, GI issues, and in the older populations, the muscle weakness, myotonia, the cardiac problems as well. And in terms of the pathogenesis, we've already covered that today. I will put this slide up here to talk a little bit about the mechanism, but not go through the mechanism of DM1, as that's been covered. But AMO-02 itself is a GSK3 beta inhibitor. We do know that in patients with DM1, they have an increased activity of GSK. This would be working upstream of the DMPK gene, and so we know that AMO-02 corrects that increased GSK3 beta activity. However, and we do have publications that will be out shortly on this next section, we also know that AMO-02 will bind directly to the toxic RNA, to the CUG repeats. When it binds to those, and inactivates them, and it degrades it within the tissue. So we believe that AMO-02 has a dual mechanism in terms of addressing two things that we see going on within DM1. We have been, believe it or not, doing clinical trials in the DM1 space since 2016. So in 2016, we ran a phase 2 open label proof of concept study with the University of Newcastle, where we did 2 cohorts of 8 patients, and the first cohort received our 1,000 milligram dose, second cohort received our should say 400 milligram dose, there's a typo on here, sorry. And all 16 patients completed 12 weeks of treatment. Nobody was withdrawn. We didn't have to do any dose adjustments. That proof of concept study and the outcomes that we looked at in that study gave us the reasons to proceed forward into a phase 2-3 double-blind placebo-controlled trial, and we were going to run this trial in children with congenital myotonic dystrophy age 6 to 16. We ran this trial globally. We had great ambitions to start this in April of 2020. As you can imagine, it started just a smidgen later than that, but we did start recruiting in spring of 2021. And over the course of the next 2 years, we were able to recruit 53 patients globally, of which 50 patients completed a 5-month treatment with AMO02 or placebo. They were randomized 1-to-1 randomization. We did have 3 withdrawals during that trial, 1 for an adverse event and then 2 patients withdrew consent. But 50 patients completed that 5-month of treatment. And for those 50 patients that completed, they were offered an open label extension program to roll over into. The initial ideal with our open label extension was that we were going to offer 52 weeks of open label additional treatment, and we have since extended that to be open-ended. So of the 50 patients that were eligible to roll over into the open label treatment, 49 of those did. And then during the course of this open label treatment, which started, that open label study started in the tail end of 2021, we have also been able to open it up to some treatment-naive patients. These treatment-naive patients were either adults with congenital who don't often have a place to land in clinical trials, or folks with childhood onset myotonic dystrophy who, again, don't really have a place to land in clinical trials at the moment. And so we thought we'd be able to give them an opportunity to enter an open label data study and get some data on them as well. So of the 50 patients from the original double-blind placebo-controlled trial, we enrolled 49, and then we've enrolled 16 treatment-naive patients. This is just a quick study designed to say there's 2 ways to get into the open label extension, which is the study we're talking about today. You could either come in from the double-blind trial, and if you did that, we'd redo an up titration to get to 1,000 milligram weight-adjusted dose for that, or if you came in as a treatment-naive patient, same thing, you'd have a couple weeks of an up titration, and then continue essentially open-ended. So this is a bit of a busy slide, but let me just break it down a little bit. Anybody who's a blue line is someone who is still currently ongoing in our extension study. So some of these patients, as you can see at the top of the graph, have been included in our studies since the fall of 2021, and so they're going on over 3 years of treatment. We did have a period of time where people had gotten to 52 weeks, and we thought, oh gosh, we want to extend this. And so there was a bit of a gap between getting some of the ethics committee approvals to extend that and people having completed the 52 weeks. So the orange lines that you see on here, particularly at the top, are people who did an entire year of open label treatment. And then for the folks at the top, there was a gap before we were able to offer them continued treatment. So some of those folks decided not to come back for the continued treatment. But once that gap had been resolved, you can see that all of those blue lines moving down forward, people rolled over directly after doing 52 weeks. Anybody in a very light blue, we had one site that completed all of their patients through 52 weeks and said for resourcing reasons, we're going to go ahead and complete our participation with AMO02. So those are the 3 in the light blue. And then anybody in the yellow withdrew consent. And we do have just 1 patient in all of this time that had to withdraw due to an adverse event. So as I mentioned, I think we see great retention in this study. 49 out of the 50 originally available subjects to go into the extension study opted to do that. 46 of those completed a full year in that open label extension study, and then 40 of those 46 opted to go on into having even more than a year. And additionally, for the treatment-naive enrollees who maybe wouldn't have screened in time for our first study or wouldn't have been eligible for our first study, of those 16, we have 14 that have the potential to complete a full 52 weeks of treatment. 7 of those have. 7 of them are still in that initial phase, so we'll look at that initial number, when they get done with 52 weeks. But of the 7 that completed the 52 weeks, they went on to say, actually, we'd like to continue to get this as well. So Quick demographic slide in terms of the breakdown between congenital and childhood. Obviously, all of the patients that came from our initial study, which was recruiting congenital myotonic dystrophy, they all had congenital myotonic dystrophy. Of the 16 treatment-naive patients, however, 7 of those came in with congenital myotonic dystrophy, and 9 of those have had childhood onset. The age range for our initial study was 6 to 16, so coming into the extension study, they would have been 6 to 17. Some of them were right on the cusp of 17 when they enrolled. And then we've seen ages 16 to 27 in the congenital, which 27 is impressive given the high mortality rate for congenital myotonic dystrophy, unfortunately. And then we've seen ranges of 6 to 16 in the childhood DM1s in terms of what's enrolled right now. So we're here to talk about our safety and tolerability of the long-term extension study. Now you know a little bit about how long of a term it has been. Overall, we've seen 526 AEs in 63 subjects. The majority of these have been mild or moderate. Out of the severe, only 2 were deemed to be related by the investigators. One of those was what led to the adverse event discontinuation. And in terms of potentially life-threatening, that was unrelated. Most of our AEs are unrelated. We do have a handful that are related, and I'll give you a breakdown of these in a second. And then these are kids that have, serious medical problems. So in terms of the SAEs, we have had 31 occur over the course of the nearly 4 years in the extension program, but I think that's to be expected given the acute medical care that these kids get on a routine basis. I realize there's a lot on this slide, but let me just highlight that this is any treatment emergent AEs that are occurring at greater than 5% incidence in this extension study. And for the most part, these are what you would expect to see with school-age kids. I have a 7-year-old. I think I've seen most of these in my household. And the ones that I've flagged here are AST and ALT elevations, which again, Dr. Lilliker mentioned. This occurs sort of naturalistically in this population as well, but it is something that we expect to see with AMO02, so let me break that down for you a little bit more. In the extension study, we have seen over the course of this time and this many participants, about 35% of participants, so about a third of participants have had at some point in their participation an ALT or AST value that has breached the 3 times upper limit of normal. When it happens and it gets to a 3 times upper limit of normal, that's kind of a watch and see thing. We monitor that. If it gets to above 5 times the upper limit of normal, we'll look at doing some dose reduction or changing the way that we're dosing with them and manage it that way. However, in our prior studies, and let me just say that coming into starting these studies in myotonic dystrophy, we had over 500 patient exposures, and now we're above over 600 patient exposures with sitagliptin. We expected to see about a 15% incidence of ALT and AST, so this is something that wasn't unfamiliar to us. And then I think when you add the natural disease prevalence for this on top of that over the course of 4 years, that's where you'll see some people rise above the 3 times upper limit of normal, about 1 in 3. We do know that in all of these instances, they're not co-occurring with bilirubin increases, they're not meeting Hy's Law criteria, they're not having abnormal physical exams or abdominal pain, and so it really is an ALT or AST increase that we would expect to see in this population. The ways that we manage it, like I mentioned, if it gets to a point where the watch and wait and it would continue to be something like above 5 times the upper limit of normal, at that point we'd go ahead and do a dose reduction, wait and see what happens, and then we may re-challenge them as well, or we might keep them on the reduced dose for the length of the study. But this has been something that we've been able to monitor and work very closely with our PIs on, and have had success in keeping everybody but one person in the trial. And that one withdrawal criteria, like I said, that was because they met a stopping criteria, and it was fairly early on. We did follow up with them, obviously, after they discontinued the drug, and that has come back down. What we see in these ALT and ASTs is that they come up, they plateau, but they come down. So we haven't had a case where the ALT hasn't resolved, whether that was a dose reduction or just watching and waiting, or a re-challenge and it doesn't occur again. Just to touch on efficacy, this is an open label trial, so keep that in mind. However, this is being rated on a clinical global impression scale from the time that the participant entered the study. So if they entered the initial double blind phase, it goes all the way back to their enrollment there. If they entered as a treatment naive patient, it would go back to their experience in the open label trial. And I'm sorry about the colors. I will talk with the programmers because A, if you're colorblind, this is a horrible slide, and B, they're boring. So I'd like to see something a bit more jazzy on the colors, but essentially, let me see which side. The brown line means no change at all. Anything to your left of the brown line, I have to actually figure out which side that is, would be either minimally, moderately, or improved. Okay, so anything to the left is going to be improvement. Anything to the right of that is going to be a worsening. And this is a 7-point Likert scale with 4 right in the middle. 1, 2, and 3 are improvement. 4, 5, 6, and 7 are worsening, and you can see that even on here we don't get anything greater than a 5. So the meta message on this slide is really that the majority of participants have shown clinical benefit in the categories or met a not change even after years and years in the study. So I think from an initial efficacy, it looks like patients are staying in the study, and I would hope that it's because this reflects why they're staying in the study. They're feeling like they're getting some clinical benefit. And just an overview, we've had excellent day-to-day tolerability. This has been our experience throughout the entire program. We don't have any concerns. I think it's reflected in high retention rates as well as the favorable CGI scores. The one thing that we do manage is LFT elevations, which again is expected for this population, also is expected for this drug. But we've been very successful in monitoring and managing that in a way that keeps the kiddos in the trial. We do have DSMCs that have been convening on these trials since 2016 on a quarterly basis. They've never flagged up any changes that we need to make. And we obviously, on a routine basis, have discussion with regulatory agencies on our progress. So thank you very much for the opportunity to present today. Obviously, a big thank you to study participants and their families. It is no small feat to get to sites. Our site staffs and collaborating investigators have been fantastic. The DM1 patient community has been fantastic. I do have to run and catch a flight, so if you've got questions for me, feel free to email it. Check out our website for upcoming what we're going to be doing next. And then, not to outdo PepGen, but this is our entire team. We thought we'd add our dogs